Hello, we are taking a look at the digestive system. We're going to focus on the small intestines. We'll start with the small intestines. The small intestines is made up of the duodenum. I know it's pronounced funny, I had to look it up too. Duodenum, jejunum, and ilium in that order. Ilium spelled with one L and one I, not an E. No, I messed up. It's spelled with an E, not an I. Ilium with an I was the bone. Ilium E is the small intestines section. So it goes duodenum, jejunum, and ilium. Where do we find them? You can best see it if we remove the liver. The stomach connects to the duodenum. So you can see a piece of it right there. That's the duodenum. Let's remove the stomach. From the stomach to the duodenum, you go through the pyloric sphincter. The duodenum has connections. The pancreas releases pancreatic juice through the pancreatic duct into the duodenum, and then the liver releases bile through the gallbladder and the bile duct. So in green, that little tube that's cut off there, that's a bile duct into the duodenum. And they share a passageway, the sphincter vati, the sphincter vapula, leading into the duodenum there. All right, so that's the duodenum. It's just this C-shaped piece. It's retroperitoneal. It's the C-shaped piece right here. Just that. But it's pretty big, and it's really important. Obviously, you're getting lots of food, chyme from the stomach, getting bile from the liver, gallbladder, getting pancreatic juices from the pancreas. The duodenum then continues on peritoneally, anteriorly, to the jejunum. So it's kind of circuitous here, but it starts here, wraps around, and whatever, keeps going and wrapping around. I can't really point out specifically where the jejunum starts and the ilium ends, or jejunum ends and the ilium starts, but jejunum, then ilium, and then the ilium ends right here, leading into the cecum of the large intestines. So here's the last part of the ilium. You can see the ileocecal valve, the sphincter leading from the ilium to the cecum. So, what characterizes each region? The duodenum has a decent amount of villi, and let me back up. What you can see with your own eyes, these ridges, those are plicae circularis, and that's just to increase surface area. More surface area means there's more room for digestion, more room for secretions, which we need both of, and absorption, which we need all of in the small intestines. So at a gross anatomy level, with your own eye, you can see plicae circularis. At a microscopic level, tissue level, you can see villi. So these are villi here too. These are villi. So there are some fairly prominent villi in the duodenum. The villi are most prominent in the jejunum. That's where most absorption happens. Most chemical digestion happens in the duodenum. Most absorption, nutrient absorption, happens in the jejunum. So you see the most prominent villi there. The ilium, let me back up. And also in the duodenum, you can start to see some glands. There's some characteristic circular glands. They're not shown here, but when we look at the histology, we'll see some glands that are characteristic to the duodenum. The ilium, what's characteristic about the ilium, this latter part leading into the large intestines, the cecum, you have these lymphoid follicles called Peyer's patches. They're very prominent in the ilium, and take a look at the histology videos for that. So that's how you can tell the difference between these different areas, both on surface features and histologically. Most of them do have plicae circularis. I know I showed it here for the duodenum just because this model is cut here to allow for me to show that, but all the areas of the small intestines have it. Generally, the small intestines have the same basic features. Like what we've seen before in previous video, looking at the layers of the digestive system, this is representing the intestines. You've got your mucosa layer, you've got your submucosa, muscularis externa, and serosa if it's a peritoneal organ. So not the duodenum, but the jejunum and ilium. They have a serosa. Otherwise, it would be just interstitial, or excuse me, dense irregular of adventitia. But this epithelial layer with the villi, this is simple columnar, has these villi to increase surface area. These simple columnar cells, not shown here, but there should be microvilli. So 3 layers, three orders of magnitude of increasing surface area. On a gross anatomy level, we've got the plicae, then we've got the villi, and then even smaller on each cell, you've got the microvilli on the apical side. Also, part of this mucosa layer, you've got blood vessels. These capillaries are fenestrated, meaning they have larger pores than normal. They leak more than normal because you want to get nutrients in, but they're not so big that you can let large cells in. You want to be able to get lots of nutrients, but not like large pathogens or other things that are large. Or you also don't want to spill out your own cells. So you can find fenestrated capillaries here for absorbing most nutrients like carbohydrates and proteins, amino acids. But you also have these vessels shown here in yellow and shown here in green. These are lymphoid capillaries, lymphatic capillaries known as lacteals, Lacteals, lacteals. Fenestrated capillaries, lacteals found in the villi to help absorb most nutrients like carbohydrates and proteins. These are to absorb fats, lipids. And you can see that going deeper. You have villi that poke out, but then you have some parts that come in. Those are called crypts. This is where we get a lot of mucus production and also regeneration of cells, because these cells, they die quickly because they're exposed to things from the outside. Often you'll see in between these simple columnar cells, you'll see cells that are a bit lighter in color. So we're zooming in here, and you can see these cells that are lighter colored. Those are representing goblet cells. Those are representing goblet cells. In this image, in this model too, you can see the ones in blue here. Those are representing goblet cells. They produce mucus. We find goblet cells all over our body. We've seen them in the trachea, for instance, in a respiratory tract. Same ones, goblet cells produce mucus to lubricate the lumen of the intestines. This is the muscularis mucosae layer. Muscularis mucosae, you find that here too. Muscularis mucosae. So that's the mucosa layer. And lamina propria is mixed in between here. That's areolar connective tissue. The submucosa, areolar connective tissue predominantly, and blood vessels and nerves. That's the submucosa. This is the submucosa. Muscularis externa. Some are circular, some are longitudinal. That's why they look different. They're cut differently. And then outside of that, if it's the jejunum and ilium, we have serosa. If it's the duodenum, it would just be dense irregular adventitia. Over here, muscularis mucosa, different layers, and then deepest or most external layer, that's our serosa or adventitia. The jejunum has very prominent villi. If we're looking at the ilium, this is a better model of the ilium because this has a large lymphoid follicle. This could be a Peyer's patch that's dipping into the submucosa. Here's a germinal center of that lymphoid follicle. All right. I believe those are all of the important features that I'd like you to know of the small intestines. Remember the order: duodenum, jejunum, ilium. DJI. Duodenum, jejunum, ilium. They all have plicae circulares. They all have villi. They all have microvilli, just to different extents. The jejunum has the most amount of villi and plicae, because this is where we're absorbing the most nutrients. Ilium has prominent Peyer's patches. Duodenum, it has these submucosal glands, which unfortunately these models don't show. And in this model, this is just generic small intestines showing the same layers that we've seen. Here are the villi of the mucosa, muscularis mucosa, submucosa, muscularis externa, serosa, or adventitia. All right, thanks for watching. Please let me know if you have any questions. Thanks and take care.